So today is the closure of your webinar series, right?
Yes, sir. Yes, sir. No, he is just making the live stream. So
today is the closure of your webinar series,
yes,
right?
yes, sir. Yes, sir. Yes, sir. No, he is just making the live stream. So
today is the closure of your webinar series, right?
yes. So welcome all of you, sir. Our speaker is here.
Can you hear me?
Yes, I can hear you, sir. We can hear you clearly.
Oh, okay, because there was some music, like I don't know whether music comes from.
It is from this side, sir.
Oh, okay.
This is our PowerPoint about our Covinar. Yes, sir. Now our Dean sir is here. Sir, actually, he's Professor Muthumani from Vista Institute. He's
Okay.
Professor Mathur is our Dean, School of Life Sciences.
Good morning.
Hello,
to
sir.
you. Good morning to you, Professor Muthumani. And though
Thank you.
this evening with us, but I know that the time difference, and I'm extremely thankful to you for accepting our invitation to be with us today. And I know that your contribution is extremely, though I'm not a person of infectious diseases, but I could see that you have a lot of contribution on infectious disease, and you are director of lab of emerging infectious diseases, and you are with the Vista Institute, Philadelphia. And at this point in time, I'm sure that you would be associated with the vaccine candidate developments for COVID-19. And so I think this talk would enlighten all of us with the lessons which we can learn, and we can see your experience. And I would definitely like you sometime to visit Pondicherry also after COVID-19 and be with us. I hope you must be coming from nearby areas only somewhere.
Madurai. I am from Madurai.
So it's good. So then I think next time, whenever you please try to make a schedule of visiting India, you please try to visit us in Pondicherry as well, and that will be a great stimulus to all of us for establishing some kind of collaboration with you. And certainly, we would be looking forward to your presentation today. Thank you so much.
Thank you.
Dr. Muthumani, why don't you introduce the speaker and then we start.
Yes, sir. Yes, sir. Yes, sir. Let me formally welcome everyone. Yeah.
Thank you, Dr. Mathur.
So good evening, everybody. I welcome you all to the final interesting session of a week-long international webinar on COVID-19, Covinar, organized by Department of Biochemistry and Molecular Biology, Pondicherry University. Today we have with us an expert researcher in the field of virology and immunology, Professor Muthumani, Director, Laboratory of Emerging Infectious Diseases and Associate Professor of Vaccine and Immunotherapy Center, the Vista Institute, Philadelphia, USA. We are very fortunate to have you here, sir, and I extend a hearty welcome to you. I would like to add more about our distinguished speaker. Professor Muthumani directs the Translational Research Laboratory at the Vista Institute. His key areas of research include molecular virology and immunology with focus on synthetic vaccines and immunotherapy. He completed his postdoctoral fellowship from University of Pennsylvania. He has been working on vaccines and immunotherapy for last 22 years and has received several honors and awards, including Young Scientist Award at the Nobel Symposium on HIV, Young Investigator Award by International Cytokine Society, Young Investigator Award from International AIDS Society. He has received several prestigious grants from NIH and has been awarded 22 patents. His lab is involved in prestigious clinical trials like first clinical studies of synthetic DNA vaccine for MERS coronavirus, the first in man Zika vaccine clinical trial, etc. He published more than 100 highly cited papers in prestigious journals like New England Journal of Medicine, Nature Cell Biology, Lancet, etc. His main research contribution includes developing synthetic vaccines for HIV, chikungunya, dengue, MERS, Zika, Mayaro, and of course the ongoing COVID-19 virus. So we are very honored by your presence here, sir. I kindly request you to give your address. Thank you very much.
Thank you. Good evening to all. First of all, thanks for the invitation as well as the introduction. Really great. I think everybody can hear me well. If not, please raise your hand because since this is like a virtual meeting, it's not like in a in-person meeting, right? So before I want to jump into that, right? This is a presentation. Is a purely, I put a disclaimer here in the bottom, it's purely publicity data from the internet or like some other research source, like I'm compiled together to educational purpose, like a knowledge sharing, right? I'm not criticizing any of the data, whatever the data is there, published, I'm going to talk about to give some example, how we moving forward towards the vaccine and the therapeutic intervention against the ongoing, the pandemic COVID-19, right? So it is not I'm, even though I'm representing for the Vistar Institute, but not the presentation, nothing with Vistar. So purely my personal interest for educational purpose. So keep in mind, right? So should not be shared or photocopied and distributed. Sorry. Okay. So just I want to give you a background information about the Vistar Institute. Pretty much most of the biomedical researcher, we know that it's one of the best or first vaccine center developed for in the entire world. So, for example, I can give you an example, the rubella, one of the MMR vaccine combination was developed at Vistar in 1969. And also the rabies vaccine, the two of the rabies vaccine, which is also developed from the Vistar Institute, back in days, right? Currently, the millions of children's, throughout the world, benefited because of this rotavirus vaccine, which is also developed from the scientist here at Vistar. His name Paul Offit, right? Still he's in a faculty at Vistar Institute. And also recently, the first human vaccine for against Zika virus also developed from my lab under the leadership of David Weiner, my mentor and my boss and like a chairman of my department. So that all being developed in this Vistar Institute. You can see that the top panel is the old building, original building. This is the current floor. You can see that. And also you may know that a first animal model, a Vistar rat. This is the statue of that. So it was developed or generated at Vistar Institute. So such way the Vistar is such a prestigious institute where I am from that institute. Okay, so I presented this one several times, but just I want to emphasize more on that. There is a huge dynamic changes recently, particularly the emerging infectious disease. 20 years back, we may know one or two viruses, right? But now, if you look at that, we are hearing different viruses. For example, 2006 and 7 chikungunya throughout the even though it was discovered in 1954. But 2006 and 7 is a huge outbreak in India and Asian countries now, in South America and even including throughout, become now a global travel disease. Same thing in 2002 the SARS outbreak happened, which we can talk later. That is but even though that was contained only in China, it's not really spread outside of China except in Canada. Some cases have been reported. In 2012, like, you all know that the second version of SARS, which means like a Middle Eastern, another type of coronavirus, which I'm going to show some data from our lab, which we developed for vaccine against that. It is like a Middle Eastern respiratory syndrome, like, had a very impact. The mortality rate almost like a 35%, which is like a significant death rate. But the government and the international organizations, right, really worked very well to contain the infection, particularly in India also. Same thing like Zika, you all know that like 2015, 16, like a huge outbreak and become like it's a globally like a very big concern. So now still like the South America is ongoing. So the dengue also always be there. Nipah, like another emerging infectious disease, like 2017 and 18 in Kerala. I think 18 Kerala, it's become. So there was a always dynamic changes, like the virus is always evolving or coming in a different way to attacking human being. So now like we are encountering the COVID, like SARS-2, which is we going to talk about that one. I'm sorry, that's not. So for example, if you do what is like COVID, this is the first picture which I took, like even though it's say February, that time this virus is only issues like I mean China, right? So that time the scientists like our group like we had a chance to get the information that there is a new outbreak in China. That time there was a January. I'm talking about January 10th. So only we thought like it's going to be only within the China, but if you look at the today date, like throughout the entire world, like global, like everywhere, like there is a huge. This is the picture taken from WHO. That was a report according to yesterday. So see that like pretty much everywhere, like this virus, particularly, it doesn't matter. It's a developed country or developing country or underdeveloped. It doesn't matter. Everywhere it has been attacked, like throughout the world, particularly like US and South America, like all the Russian candidate, European, India, like become. It's a big problem. So this is a report overall, like you can see the numbers of cases being reported globally according to this is the data from the yesterday, and also like more than 6 million deaths, like which is like that's why it's considered as like a pandemic. You all experienced like every last 3-4 months, we all like going through, in not only in India, everywhere in the world, like people are suffering financially, economic, like financially, all kind of like suffering because of the still ongoing issues. But if you look at this is the profile of, right, particularly America, like if you look at the yellow curve, like see, but if you see that like this is started slowly like in April, but keep going up. This is not like sustained, like it means it's coming more cases. But if you look at the Europe, like if it is the green, yellowish green, like you can see that anybody can see my arrow presentation, right? Hello? My mouse, can you see the
Yes, sir, definitely.
Yeah. Okay. Yes. So this Europe, like see, even though there was high cases, but suddenly they actually took a very good measurement. But now, like there is slowly going down. But this is the like the purple color, like which is like Southeast Asia, including India. But even though we started late, like India started late, but it's now getting bigger and wider. So this means even in the Mediterranean, like the Middle Eastern areas, so like that cases also been reduced. Africa also getting bigger. So this is the really a concern. So we need to be very careful how to stop this curve, like how to reduce the curve. So that's the biggest like challenge we are facing right now. So what is meant by this virus? Why we care about? So basically the SARS, right, is one of the like a major new disease like 21st century. In 2012, like there was a lot of cases being reported in China, which I mentioned before. Some few cases also reported from the people who travel from China to America, like they got disease. But luckily, like we don't get other countries like not get that disease. So then, like it's also in overall like cases versus the death rate is small, so we completely away from that infection. So again, I'm coming back to the same thing. Like now, we are only talking about the emerging viruses, right? But there's not only viruses issue. Like there's like several type of bacteria, parasites, like a fungus, like those all. The question is $1 million question is what we have. How do we respond to different pathogens? Do we have any common platform? Do you have any common technology to respond, right? Not only that, if we don't know that any new pathogen comes, how do we can overcome? So that's always like a challenging for a scientific community, which is right now we are facing that COVID situation where people are dying on another side. Scientifically, we try to make a vaccine or intervention. So a lot of are running a race, like a lot of people are going, it's still ongoing on that issues. So how can we control this, right? So in terms of infection, right? There's a two-way we can do that. Only like a prevention, right? That which comes under really like in a vaccine or some kind of monoclonal antibody therapy. Those kind of things, prophylactic monoclonal antibody, we can be able to control the infection. That's called prevention, right? Another one is called therapy. Therapy means like in therapeutic means like after you get infection, we can treat the disease, right? But if you look at the other side, on the left side of the chart, you can see that. So it could be that any infectious disease, it could be within the region. Region means it could be a human, animal, the transmission, right? So that always been for example, swine flu is the best example. Also, same thing like a community, also same comes under category. Then population, which is like human to human. Right now, the COVID is a typical example, right? So individual cost. So for example, individual person can transmit all these factors. So this is always is kind of cycle to particularly where you stop. How do you stop? That's the important, right? Because once if your individual person is being infected, if you stop them, then you can avoid the population infection, right? That population also, if you stop the population, then you can stop the community infection, then region. So now become a global, so this is perfectly fit into that, from single location, single region, it become a global pandemic. So in terms of virus or any pathogens right now, so always you have to look for two type of responses, right? One is called natural immunity, always be there in your body, right? But the two response, which means it's an antibody mediated immunity or T cell mediated immunity, right? Natural immunity always be there, depends upon the individual, it may respond, may not respond, right? For example, immune compromised patient, they may not have a natural immunity. Whereas sometimes you have good immunity, you can be able to restrict the viral infection. So, but the thing is when you get an infection here, actually, immediately you will get the viral load. As soon as your natural immune system, defense mechanism, kick on, that means then what will happen suddenly, virus will drop, right? The viral load will drop. By the time, your T cell also helps them to drop this viral infection. At the same time, you also develop the immunity. But the long-term immunity comes from based upon these two T cell and B cells will help them to sustain or retain the long-term immunity, which is important. That's why a lot of vaccine is mainly focusing on developing this immunity, antibody immediate immunity or T cell immunity. And also they want to have a lifelong, a long-term immunity. So these are the important characterization for any vaccine, we are designing or developing. It's very, very important. So this is the basic immunology textbook. I mean, most of the audience may know that. I don't want to go back again, but what I'm just to emphasize one thing: immunity is a common word. That could be any way of you could generate the immunity, right? So, for example, there is a two type of immunity which we are very, very important. First one is which I mentioned before, an innate immunity. That's called first line of defense. As long as your innate immunity responded well or reacted well, you will get a defense mechanism which may be able to stop the replication your own. But if you failure or something wrong, then immediately as soon as you will get adaptive immunity, which means second line of defense. That's called T cells, B cells can come play a role, right? Whereas in the immune system, recognize that naturally you can get immunity, right, or artificial. This is the place where I'm talking about the vaccination will help them to induce your immune system, right? So naturally, some people can develop immune response, which could be very good. That means by active infection or maternal, they will get the immune system. Whereas artificial means a vaccine or therapeutic intervention, which help them for both way, antibody transfer or immunization. So these are the simplified mechanism. I'm trying to explain that how we can generate immunity in our body. So the vaccination is nothing but it's a training yourself. Like a training means like, you basically tell them or give the instruction to immune system to benefit for your own, right? For example, that could be a immune system has like a lot of things like thyroid, thymus. Those are the immune cells, like highly, right? So when you immunize, like with a vaccination, like in a process, basically that can leads to the like an antigen. Antigen means the foreign protein, like I consider as a like foreign protein means is against our. I mean, it's new to our immune system. So by doing that, like you basically that immune system will generate different type of like response, including cell-mediated immunity and also like antibody, which is called like a polyclonal response, which could leads to the perfectly very nice way of like neutralization. So that means these T cells like could generate with the help of that B cells help and T cells help it together. You get a good amount of like a good neutralization antibody as well as memory T cells. So that's why you are basically train your immune system by vaccination. You are getting like your immunity against particular pathogens, right? That's called considered as like a vaccination. So what is coronavirus? I'm coming back to the basic, like now I'm coming back to the coronavirus, right? So what is the coronavirus? So I think you guys in the last 3-4 months, you've been watching a lot of medias, like newspapers and online. You know that familiar with that, right? So it's a common cold, but it's a severe to life threatening, depends upon the infection. So it could be a mild, it could be a severe, it could be a dead. Again, it's a respiratory disease. That's why they called MERS, SARS, like this, right? Severe acute respiratory syndrome. That's what the name given. MERS means Middle Eastern Respiratory Syndrome. So they all comes under like coronavirus. The coronavirus have like a different family. So right now we are talking about the novel coronavirus, which is like fall onto the same thing, beta coronavirus. So this is the picture which you are seeing here, actually taken from the NIH website, where you could see that infected cells stained for the green color, whereas these viral particle, like which is like look at the crown structure, where you see that this is the computer prediction, whereas you can see that nicely this comes out of the cells, are decorated outside the cells, or trying to get into the cells. You can, right? So this is the way that the virus infects the cells. Where it came from? So this is again like it's $1 million questions. Like I mean, still we don't know the origin of this COVID-19, but we know very well that origin is bats is the the host for most of the coronaviruses, including like SARS-1, and also intermediate host like which called a civet cat, like which is palm civet cat, which is like spread mainly by the SARS-1 viruses, which you can see that in the picture. Second thing is like the 2012 and 2015, like there was another virus. It came from the bats to camel. You may ask a question: how did camel got there, right? If you look at in the desert, like in the date plants, like the bats actually try to eat the saplings, like I mean, eat the fruits, like and then they put that fecal matter, like it can be thrown into, like I mean, not throw means like basically in the water contaminated. The camel can drink. That's why camel got to become a receptor called DP4 receptor. Then in the Middle Eastern, like there's a highly association with the human and the camel, like because in the Middle Eastern, that animal is considered as like one of the household animal for like a farming, milk, meat, everything they use. That's why this virus has been spread to bats to camel, camel to human. The third one, which is right now, is like in China, like which is the Wuhan, like where still is a questions, but like based upon the sequence homology, we can say that pangolin, like is that it's an intermediate host from the bats to human. So because they eat like pangolin, so maybe they get the disease from the pangolin. So still it's not been confirmed. There's a lot of studies going on, but we will know by sooner or later. Okay, in terms of type of vaccine, there's a lot of vaccine which is like I don't have time for going through everything, but today like my talk is mainly I'm going to give you an example from the published data. What does mean by RNA vaccine generated by Madonna, and also I'm going to give you data for the viral vector, which is adeno chimpanzee adeno chart adeno from the Oxford data, from the clinical data. Third is like I'm going to give the Innovia DNA vaccine, which is also developed from our institute. So I'm going to talk about their published data. So even though there's a several platform is been available, like now, for example, the whole virus, live attenuated, subunit, VLP, then viral vector conjugated. So today I'm going to talk about more on the DNA, RNA virus, RNA platform, as well as the Oxford vaccine platform. So the first data, like you all know that you've been also watching the TV all the time, like whereas in US actually spent a lot of money on this Madonna, which is RNA platform. So what they did actually, this is the data which I'm going to show you the clinical trial based on clinical data. But before that, I want to give you an example. Yeah. So here's so what they did actually, they used so like SARS-CoV-2, like COVID-2, they called it coronavirus, right? So there is a called crown-like structure which is made up of a spike protein, which is a surface protein. So that spike protein is the one who attached to the host cells as soon as you get into the body. So that's the way they used like a receptor called the ACE2 receptor, where they try to dump the genome into the host cells. Then they replicate and come out of the cells, which I showed you the electron picture, right? So what they did, they utilized the same like a technology. They used that spike protein, particularly perfusion trimers formation. They did it with RNA, right? So RNA and formulated with like particle. So that's why they used as a vaccine candidate. And they did with all the preclinical data from the animals, not animal laboratory studies, as well as small animal studies. Based upon that, they got a funding from government organization. Then immediately they started a clinical trial in I think 2-3 months back. Yeah, April sometime. I don't know the exact time. So where they enrolled, I think pretty much like a total of 45 volunteers being enrolled. So keep in volunteers, not in these are naive people, right? Naive. Then they had a group from the male, female, age around like average of like 35, mostly young age group, right? And within the group, actually, they had all the ethnic group like American, Asian, Black, White, all other Hispanic and thing. So pretty much like and also like they had a average of body weight and everything you need to consider in order to enroll them. So they enrolled like a 45 volunteer, and the data published like 2 weeks back, which I'm going to show you here now. So this is the Madonna adverse effect. Adverse effect means when you inject any vaccine, so the doctors, like the physician, actually make sure that the volunteer get any disease like any fever or any kind of pain, things like that. So the pretty much like you can see that this is they called like a mild, moderate, and severe, where like only few cases been reported for the severe, but most of them some kind of like mild symptoms, like not really bad. So some cases, always be there like systemic symptoms like a fatigue, fever, some headache. That's a common, very common. And also 100% after the second vaccination, especially the higher dose group. So only the first dose, they didn't get anything. But after second immunization, they got some kind of symptom, which is not life-threatening. So that means this safety-wise, this is not bad to the volunteer. In terms of the two immunological response, which I mentioned, right? So one is the T cell assays and the B cell assays. Particularly this kind of viruses, like neutralizing antibodies, is very, very important and very important. Not only important, it's essential also, because that's the way you can stop the entry, because the viral neutralization is supposed to block the viral entry. So here is the antibody data which they showed they tested like in 3 concentration: 25 microgram of vaccine, 100 microgram of vaccine, and 250 microgram vaccine. But they see that even in a small dose also they get a good response, but overall the high dose also a little bit higher, but not that differences, right? This is the data where they're comparing with the convalescent patient. Convalescent means like people who already been infected with the COVID-19, so they used as a control serum. So that means what they're trying to say that this is the people who recover from the infection, right? Keep in mind that's why considered. So that means this is the level of antibody is necessary for clearance, which you can see that's what they're claiming that this. Then also they did this is the full length protein. Then also they did the remember the spike called RBD domain, which is the receptor is AC2. So they did it with the binding domain region, where again you can see that the nice correlation, which means it's a good binding. Then they did a neutralization assay, right? So neutralization assay is the way you can measure that antibody can be able to neutralize the virus. That's the readout, right? So where you can see that again, 25 micrograms, 100 micrograms, 250, now they're showing that a good amount of neutralization. But the convalescent patient, have a huge variability from here to there. So it's there's a changes, right? But when they did it with the live virus, this is the pseudovirus means it's artificial virus. Keep in mind, only they use spike protein. The rest of them is different. So that's called a pseudovirus. So basically, it's mimicking the viral, but it's not really a SARS virus. But this is the real virus, which means the original SARS-2 viruses, where they tested only in a 25 and 100 microgram. So this is the convalescent patient data where they are claiming that they have a good neutralizing titer, which means significant number. What about the T cells? Remember, I said it's not only important for the antibody, T cells are also important, right? But this data, from this data, they are showing only they were able to get a good amount of, not good amount, is some level of CD4 cells, but not really CD8 cells, which is it's really it's not good, because T cells, CD8 cells are important for the clearance, which is in a viral, even though it's of antibodies important, in order to clear T cells also very, very important for the any viral clearance, as well as the elimination of the viral particle. So you can see that nicely, they have a good amount of T cells, but unfortunately not the CD8 cells. Okay, that's what the data is promising from the clinical studies. What they're saying that they were able to induce a good immune response based upon the data. Now they're actually moving to a next phase, right? Because they're claiming they feel that this vaccine, that the data, that antibody they generated or got from the human trial is enough to stop the replication. That's why now they're going phase 3. So next, last 1 week, you've been know that from the media, and everybody's talking about the vaccine, Oxford vaccine. What is Oxford vaccine So the one I presented before, it was the RNA vaccine, just RNA, only small fragment of this RNA, right? Keep in mind, spike protein. Here, what they did actually, they used chimpanzee adeno. Chimpanzee adeno is a common adenovirus, which is pretty much there's a several serotypes. But you cannot use a human adenovirus for the vaccine purpose, because we all exposed to adenovirus. So that means our immune system already pre-existing, that immunity is there. That means you may not be able to get the transgene expression. That's why they use the chimpanzee. Chimpanzee is close to our human family. They use chimpanzee ad. That's why the chim ad. They clone into the spike protein into this vector, and then they used that as a thing. But what they did actually, this group actually did a phase 1 and phase 2 studies. When you talk about the clinical trial, means normally they go a phase 1, which means safety, a small number. Then phase 2 or 2-1B, they extended the number of candidate, which means from 500 to 1,000, depends upon the financial support from the pharmaceutical company. Then they go for the efficacy. But what they did actually, this group, actually Oxford group, they did phase 1/2 means they did more than 1,077 volunteers being enrolled for these studies. So with support of AstraZeneca and all government funding and all those things, that's why they started this vaccine, that big level. So from that data, what you're seeing here actually, this is a ELISA titer from different thing. This is the prime. Prime means 1 injection. Boost means 2 injection. Keep it prime, boost, right? This is the convalescent, means infected people. So again, what they're trying to show that this is the data from the Lancet published the data, where we can see that they're saying that they're getting a good amount of response, antibody response, even day 1 injection. Day 7 is not that level, but day 14 they're getting good response. Day 28 is increased, but and also this antibody has been staying longer. But when they boost, they don't see much difference, still the same level of that prime. And also this is equivalent to convalescent patient. That's why they're saying that this is This is just comparison how great their vaccine is. And also they did a neutralization assay in a two different way. They did prime data where you don't see much of the neutralization. There is something it's not that great level, but they're saying that it's good at neutralization. The boost also not that level. You see that this is the level their error bar is. So this is the level, right? So here is the titer. Whereas when they did it with the other kind of assays, where also they saying this, they're claiming that that level is increased, neutralizing titer here, and also they're saying that neutralizing titer is increased. But it's a different time frame. They're saying that that antibody has been a good amount of antibody because they used a two different. This is the control vector. So what they're saying that from this data they were able to generate a good amount of antibody, even a single immunization, but bright boost also not really increasing. But they're claiming that the point is they're trying to claim that a single immunization is enough to induce neutralization. That's what the whole point is here. And also they did it with the pseudovirus, which remember the same thing I did before, because there's a lot of mutation is going on. So they used that kind of assays to generate that vaccine, where they're saying that look at the titer is average of this titer is pretty much 600 to 700. I mean, not 700, yeah, that's 500. But it's overall the time is not really increasing. But this is the convalescent patient they're comparing. That means their level is similar to the convalescent patient. Means what they're trying to say that this antibody, I mean, this vaccine is able to induce a good amount of neutralizing antibody. What about the T cell assay? So remember previously I said they don't get the good T cells from the Moderna RNA vaccine. But here this is the control vaccine, which is completely different non-specific vaccine, response. Whereas when you vaccinated with the prime, a single immunization, you can see the day post injection, right? So they're saying that there's a good amount of T cells being. This is the ELISpot assay where they measuring the interferon gamma production, which means antiviral factor. That's what T cell assays. So for example, if you CD8 cells producing a high level of interferon, means your T cells are greatly enhance the antiviral factor, right? That's what they're trying to say here. They have a good T cells, but and the boost does not really help them. Because see that average But the thing is, keep in mind, in this prime case, they have a lot of cases. Look at the number of participation, right? Whereas here only 10 participants. So only they limited for the boost. Still, they didn't say why, because they only boosted with only 10 people, but they kept only for the prime all the volunteers. So still, they didn't say in the paper actually why they stopped that boost. But they're claiming that there is some response. In summary, actually, even though those two vaccines right now everybody's talking, in addition to that, there's a Chinese company called Sinovac also producer data, and these are the company Biochemistry from the German group with the collaboration with Pfizer. Also RNA vaccine, their data has also been presented. But all these vaccines been highly particularly these two vaccines everybody talking about that. But see that initially there was 140 vaccines been preclinical trial, then all 10 of them moved to phase 1. Right now only 3 of them moving forward. So far I showed 2, now I will show another one. So in this, from these 2 vaccines, clearly they're showing that there's a systemic adverse effect, which is a common for most of the vaccination. They are claiming, both group claiming that they have a high titer of binding as well as neutralizing antibody, which the data is showing that data, supporting the data. And also they're confirming that pseudovirus and also RBD timer, which is really good. Live virus also, they showed that there's a high neutralizing titer, right? So, and also the T cell responses there. So, okay, so now I'm coming to the another platform which we've been working a long time, DNA vaccine, synthetic DNA vaccine. So under the leadership of David Weiner at Wistar Institute, he's the chairman for the Vaccine and Immunotherapy Center, where I've been working with him last 22 years when I started my postdoc. So, sorry, we are using a synthetic platform for generating immune response for several pathogens. Right now we are the first 2 vaccine which I mentioned, Zika, MERS, which actually is a first DNA vaccine. It's been successfully translated to human clinical trial. So not only that, today I'm going to show some data about how can the DNA vaccine can also be used for therapeutic intervention, which I will talk later. So the picture which you are seeing here actually is a rabbit muscles where the transgene, GAP, is a green fluorescent protein, how effectively able to deliver with the help of electroporation. So that means your foreign DNA, foreign genes can be delivered very well into the muscle cells. Second thing is that any kind of this advantage of DNA vaccine is a large synthetic protein. It can be easily manufactured within a short time. There's no immune response against to the backbone, and also you can also the DNA can multiple injection because DNA vector does not cause the immunity and viral immunity. So and also the main thing is the other two platform which I mentioned that they need a cold chain, but DNA you don't need a cold chain because it's a temperature stable. DNA in a room temperature can stable without any problem. So that's the advantage of this vaccine. So I'm going to show the data from before I jump to our COVID vaccine platform, I want to show the data from the Middle Eastern respiratory syndrome, where I don't want to say again now how bad it is. If you can see that now almost a 35% mortality rate, then means you think about that how bad it is, right? So it's really a dangerous disease. It was reported in Saudi September 2012. So immediately we designed a vaccine and we did all this preclinical data. So this is the map, world map as of 2019 April so a lot of cases been only reported during the early time period. Then there was a 2015 little bit spike happened. After that some cases still going on off like that, but not many cases. So it's still in the low level. But the interesting thing is actually in South Korea, one of the volunteer, not or businesswoman who came from South Korea and transmitting the disease to Korea. So a lot of cases have been reported, almost 1,800 cases been reported. So this is the photo, single donor travel from Saudi to Korea, able to transmitting this virus hugely so overall 36 cases been death 186 cases been reported. But South Korean government took a very good measurement, they stopped their application immediately, contained their application. So what we did actually, I don't have time to go all my preclinical data, but the one highlight data I want to show them. This is the X-ray which you are seeing here actually from the monkey who got vaccinated with our synthetic DNA, and then challenged with the virus. So basically we did two vaccination, so two immunization. After that, we took this monkey into the BSL-4 pathogen level laboratory. Then we infect the monkey with the virus. You can see that this is the control vector. So you see the lot of inflammation in the lung, whereas you don't see the much on this X-ray from these two monkey, low group as well as high group. But you can see that the graph where the viral load from the after day 6, we did the viral load. You can see that our vaccination almost a 2 log reduction. And also the histopathology, you can see in the bottom where this is the PVAX group normal, whereas low dose it's been PVAX is highly inflammatory. A lot of lungs has been infected, whereas our high dose and low dose both responded well. So almost a 3 log reduction has been treated after day 6-7. All the monkey who got vaccination completely recover from the infection. That's the paper was published in Science Translational Medicine in 2015. So based upon that, actually, the studies actually, U.S. Army actually really interested in this project because, as you know, that a lot of U.S. military personnel located in Middle East, and so they want to develop this vaccine for their use, right? So it was funded by Walter Reed, is an Army Research Center, and also Innovia Pharmaceutical, they have a technology for the electroporation and synthetic DNA platform. And the Gene One in collaboration with the Gene One, all they combined together and started this clinical trial called WRAI, right? So that's a first human trial for the MERS vaccine, our coronavirus vaccine, you can put it that way. So first coronavirus vaccine in human was this vaccine. Mainly they did it with the 3 doses, 0.65 milligram, 2 milligram, and 6 milligram. And you can see the immunization, 3 immunization. And this is the time frame we collect the samples from the volunteer for the immunological assay. So just I'm going to, there's a lot of data, but I don't want to go all the detail. So this is actually, I mean, during the course of infection in Korea, they collected the acute phase infection. That's the antibody binding data. This is the convalescent people who recover from the infection. This is the binding data, right? Whereas our vaccinated, like, a significant level of antibodies been generated, right? So that means is like, we can equivalent to like natural infection versus our vaccination. Natural infection versus vaccination, we are generating a good amount of antibody, which you can see that. Not only that, this antibody also have a functionality, which means that antibody has a neutralizing titer. So this is the EMC, like a real virus, where we can see that like this is our vaccinated group responded very well, close to the convalescent as well as the acute infection. So the statistically significant from the acute infection versus our vaccination. That means a natural infection, how this human generating the antibody, from that same way our vaccination is able to generate, which can able to clear the infection. So from this, like was funded by CEPI, which is like CEPI, which is like one of the world big organization. We are kind of funding 56 million from this platform for this vaccine, and they moved to human trial, like phase 2, phase 3 next level, which is going on in South Korea. So that paper actually, that human phase clinical trial has been published in the Lancet Infectious Disease. So now it's really highly cited. All the references based upon this data, like which is like very, very proud of saying this vaccine is designed by my group and now translated to human clinical trial. So based upon this knowledge, information which so as soon as we got the news from the outbreak, actually, January, I think 10th, like we got that night actually information about this, there's a huge outbreak. So we designed the vaccine. So you can keep in mind, like since I have a lot of experience from the previous MERS vaccine, which is similar to coronavirus, like so we immediately designed this vaccine in collaboration with the pharmaceutical company, which is like our company, and also our technology, both combined, we generate this vaccine. And this is a big teamwork where like you can see that like all the Ami Patel, like Emma, Dan, pharmaceutical help, Kate, Trevor, and like Lauren, they all like on this project, like a lot of hands in that project, because it's essential or urgency, right? So we designed the vaccine, like you can see that our vaccine is a design platform compared to the MERS and SARS. So this is structurally, you can see that our immunogen. And this is the preclinical data where I'm showing like neutralizing antibody. This data is published, like you can see that Nature Communication paper where you can see that normal mice, whereas they this is the vaccine name. So after this, like vaccinations are really worked very well. Not only the spike protein, S1 region, S1 is full length protein, and RBD. In all the cases, you can see that they have very good amount of antibody, and that antibody also have a high endpoint titer. You can see that, you can appreciate that. Not only that antibody is good, but also like there's a high level of neutralizing antibody, which is like a good number of neutralizing antibody. And the interesting thing is actually the broadly like T cell epitope is been recognized. So like that is which is very important. So that means our vaccine not only inducing a good amount of antibody and also neutralizing antibody, and also it's broadly that T cells has been broadly neutralized. Broadly generated. So based upon this preclinical data, we are a pharmaceutical company, and you start like combined with the University of Pennsylvania, they started a clinical trial like with I think 40 volunteers being enrolled. So I cannot talk about the data because I'm one of the inventor. I'm supposed not to talk. So, but the data is still ongoing. So study is ongoing, but all the 40 volunteers have been enrolled. They're analyzing the data. Hopefully, you will see the data soon. So now, so far I've given an example, 3 different platforms. One is RNA, then second is adeno data, both clinical as preclinical data. And now I want to move my gear to the therapeutic intervention. So, which I mentioned this is the spike protein, okay? So the spike protein is one of the key factor, which has a receptor called the RBD domain, which is the receptor is the ACE2. So in the antibody, any viral infection, right? But the IgM is always come first, right? Then IgG picks up later, right? So the idea behind it that like is generating antibody on this area, like, where this intermediate, like, it's a really, really important key, like within our, right? So by doing that, this, when you try to stop that replication in the time, right? So the only thing is you can do by the therapeutic intervention, right? This is the key. For example, a vaccine, when you inject them, it may not help you here, right? So that's why therapeutic intervention is a very, very important key factor for controlling the viral infection. So what we did, like, several group actually working on this, like, if you see the therapeutic drug and like, including monoclonal antibody, or some other molecule, small molecule, several people are working, like almost more than 83 studies going on. So we don't have a time for going all the details. And also, like, there's a AstraZeneca, not only is they're spending time on the vaccine, they're also working on the antibody therapy, either isolating from antibody from the patient who got infected with COVID and isolating the B cells, and they can use that B cells to clone that antibody. Then they want to produce the monoclonal antibody. So that's the idea is going on. What we are actually working on that. Oh, this is the cartoon which I'm trying to explain. This is the spike protein. From this is the RBD domain. RBD domain, and this is the AC2 receptor of the host cell. So that's the interaction. So we are trying to block that interaction. If you block that interaction, technically you will be able to stop the viral entry, virus entry. Which means if you will stop the entry, then definitely then you won't get the infection. So, antibodies, like, there's like a lot of antibodies, like, it could be like, like a millions of type of antibody, but we need to select the right antibody. So that's a monoclonal antibody is a very, very important gap. So some of the antibody which we generated, like, in the lab, like, I cannot disclose all those things. Some of them really good, some of them binding. So, like, we are also using a synthetic DNA platform. So whatever the vaccine we inject, like, we use that vaccine into mice, and we generate a lot of antibody to fill the gap, like, how we can therapeutically enter the infection. So in order to show the data, actually, I want to show that our published data from the Zika. Remember when I mentioned that Zika study is a human clinical trial? Before that clinical trial, like, we actually challenged mice after vaccination as well as monkey both. The bottom one is the, like, this is the data from the antibody data from the mouse, whereas this is the antibody from the monkey. So keep in mind this antibody actually, what I'm trying to say that this mice and monkeys been vaccinated with our vaccine, then we challenged. Then challenged from the recovery being challenged from the protective monkey, we isolate the antibody, right? Same thing like in the mice, we isolate the antibody. Then we used that antibody to use as a therapeutic intervention. So keep in mind. So that means we are taking from the vaccine, immunization with the vaccine. Then we are taking those B cells, which is like a protective B cells. Then we clone those B cells and they'll put them into expression vector as in a DNA. Then we are injecting into, like, using as a therapeutic intervention. So this is the data where you can see that from those antibody, like, we actually on day 0 we, like, inject the thing. Then day 2, right? Day 2, like, we challenge mice with virus. You see that some of them antibody, like, gives 100%, 80% infective, blocking the viral replication. Survival. This is the survival curve you are looking here, right? Some of them, like, not good, but some of them really good, like, in terms of, like, protectivity. So that means it stopped the replication, Zika replication, right? So which means, like, the antibody which we cloned from the vaccinated animal, which have that property of blocking the viral replication. That's what I'm trying to say. So how we can use this platform? How we can utilize this one to in an emerging situation. So just I want to, this is a busy slide, but I will walk through slowly. So what we did, like, we are actually working on this called rapid and persistent, two way here, right? For example, in an emerging situation, right, your vaccine may not work because vaccine required at least minimum 10 to 15 days to get immunity in your body, right? By the time your infection is like happen, you cannot stop that, right? So how do you overcome that? That means you need to stop the replication within, like, within that window, like first 3 to 4 days, you need to stop the replication. Then if you stop the replication, then you won't get the disease. but vaccine you cannot apply on the pandemic situation, right? So that's what we are coming with the strategy called the immediate versus persistent, right? So this proof of concept data, which I'm showing here, like, and also, and I published another paper in the chikungunya data, where we did actually a group of mice were 20 mice, keep it in that way, 20 mice were vaccinated with our vaccine. So we vaccinated with the 2 times, keep it, the vaccine is 2 times. And also we also vaccinated or inject our antibody clones, they're called DMAP, DNA encoded monoclonal antibody, right? That means that that vaccine will give antibody immediately in your body, keep in mind, right? So by doing that, what we did, then we challenged the mice, mice with half of the mice were challenged on day 2 and half of the mice were challenged on day 21, okay? So the data, what you are seeing here, actually, this vaccination group, right, completely failure on day 2 challenge, because the vaccine group does not have enough immunity. That's why they all dead here, right? But if you do a challenge on day 21, they all 100% protective. That means your body developed immunity, right? So this is the vaccine group, right? So that means the initial vaccination may not be able to enough for protective infection, whereas on for 20 days, you have a natural immunity developed by your body, which can you are 100% protection. Whereas in this case, if you look at the combo group, which I'm talking about, the both vaccine and DMAP group, right? You can see that even if a day 2 challenge, when I do a challenge on day 2, 80%, 90% of the mice were protected. You know what? Because of the antibody molecule which I injected, that gives the immediate protection for this group, right? See, that means this you don't need a vaccination. Your host immune system completely waiting for that immune response. But that antibody which I injected into the mice immediately systemically produce antibody within a 24 hours, 48 hours, within a 12 hours. So that's the antibodies gives the protection. That's why you are getting a complete protection or a 90% protection from the infection. Whereas later on, definitely your body, that mice have an immune response as well as therapeutic interventions, you will say, what's not moving? So you have 100% protection. So that means in the single synthetic DNA platform, combining both antibody molecule as well as vaccine together, right? So that antibody molecule gives immediate protection within a day, second or day third, you will have an antibody systemically that will stop your viral replication. Then when you vaccinate them later, you will have a lifelong immunity in your body. That's called immediate and persistent vaccination. So that's the only platform where synthetic DNA platform can be able to do that. That's what this data also supporting. We are also working on this way of for COVID also, even though vaccine is already been going on, right now we are using the monoclonal antibody therapy as an intervention for the ongoing infection. So I think this is my last slide. So basically, this yeah. So if you talk about this in the overall in the globally, this is the older slide, but still 3-way which we are focusing on the COVID problem, which means a repurposely used drug, means before approved drug for Ebola, HIV, malaria, and some other viral disease. Can we use those drugs into them for the COVID? That study is going on. Not so far, not any of those not been approved yet. Antibody therapy, which I mentioned, that it could be a monoclonal antibody therapy or DNA synthetic antibody therapy, a lot of platform is going on. The antibody also being generated from the infected people or generated from the vaccination, typical immunological monoclonal antibody generation. So you can do that for therapeutic intervention. A lot of studies going on. Third one is the vaccine. Several platform, RNA, DNA, adeno, live attenuated. Bharat Biotech, I saw last week, there was a inactivated vaccine also started. I think I read the story. There was a phase 1 clinical studies been started also. So, but the bottom line is there is a lot of progress is going on for vaccine, therapy, treatments, and prevention. So, it's a human life. Everybody's working hard, but things are very quickly we may have some hopeful outcome from these studies. So with that, I will stop here. I would like to acknowledge a lot of people because this is a combined studies, a lot of people. mainly like our team worked like here, and David Weiner, my mentor. Then Gary Gobinger, this guy is the one who like Ebola antibody therapy has been generated by him. He helped us a lot of this Zika studies and MERS studies. Pablo is our clinical director for all the clinical studies. Funding from Innovia and the Gene One, like supporting all these like our clinical studies. NIH for the Dr. Heinz Fieldman for all the virological assays, challenge studies, and all those things. Like now, without their support, we cannot even do the phase BSL-4 level pathogens, which is a risk. Like we cannot work on. So with that, I will stop here. If I am happy to take question.
Thank you very much, sir, for the spectacular talk. I'm sure that all our participants would have greatly benefited by your lecture. I would like to share here that more than 1,000 participants watched this talk live, both through this platform as well as through YouTube. Now we request our respected Dean, Professor Mathur, to share his word of wisdom.
Any questions like you need to answer?
So 1 minute, sir. I would first ask from Dean and then.
Yeah, sure. Okay, thank you.
Yes. Sir, actually, your mic is muted, sir. Professor Mathur, sir.
I'm telling that it was a very wonderful talk. I think questions should be addressed, and I don't have anything to say on this issue. And it is extremely informative, and I think all the participants would have got benefited out of your presentation. And I think we should look forward towards more such presentations to various groups of people within the country. And I think this COVID has given an opportunity for us to meet from different places, and I think we are trying to make use of that opportunity what we have. And it's really good that 1,000 participants or whatever you told, and I thank all of them to join with this, and then they will stand benefited. So please continue with that.
Yes.
Thank you.
Thank you, sir. Now the session is open for discussion. I request all the participants to post your questions through the chat box. I request Dr. Subbalakshmi to moderate this session. Subbalakshmi ma'am, your mic is muted. Yeah. Yeah, thank you.
Yeah. Thank you, Dr. Rukmani. My sincere thanks to Dr. Muthumani for the excellent talk. Participants have posted many questions in the chat box. Let us begin from Gopika. She asked whether most of the vaccines being developed for COVID-19 is targeting ACE receptor.
So question or a comment?
No, it's a question. She has asked whether most of the vaccines being developed for COVID-19 is targeting ACE receptor.
Okay, keep in mind, ACE receptor is the RBD. Like the spike protein is the RBD protein is within the spike, right? This is in the middle region. That is the receptor. Like ACE2 is the human cell receptor. That's the interaction. Like it's right. So the vaccine is generated against the spike protein, right? So RBD is also in the part of the spike protein. So you don't need you cannot make a vaccine against ACE2 because ACE2 is our host cells, host receptor, not the viral protein. So you have to target the viral protein, not the other way around.
Okay. Thank you, sir. She also has another question. Whether the animals used in vaccine development has shown symptoms entirely different from known symptoms of COVID-19.
Even I showed the human data. Why is he care about I don't want to say care about it. So animal don't show. This all the platform what I presented from the other group also it's a safe, very safe platform. Already have a lot of historical safety means highly this virus. I mean this platform doesn't cause any disease or any adverse effect to the animal because that human also doesn't have any effect on them. So no issues with animal.
Okay. Thank you, sir. So the next question is from Dr. Kesavalu. He has asked, in the Moderna data, why the immune response duration is not given when the immune response is starting with Moderna vaccine?
They did actually. They gave a day 14 to day 28. That's the time frame. Like that's the normally they can do that. You cannot do every day. You cannot take a blood from every day from the volunteer, right? So there's a time frame, like for all the clinical studies, like already been set. Like the doctors decide. So you have to tell them, like we want to have a window. Like remember, immune response you need at least 14 days to. That's the gap. Like day 14, day 28, day 56. They have a different time frame. I think they had antibody data and the ELISA data, ELISPA data in a different time. They do have the time. Yes, they collected different intervals.
Okay. So he has a couple of other questions also. The next one is, why only RBD is targeted for the vaccines? Why can't the N-terminal domain or C-terminal domain for vaccination protocol?
Who said? Who's asking? I think repeating the same question. I think I already explained.
It's like case of
spike protein is a full-length protein, so not the RBD. So just change the mind. RBD is that they're talking about the RBD is a receptor that the AC2 receptor. That's right, but they used full-length protein. Yes, spike protein.
Okay.
That's a vaccine candidate. Yeah.
Okay. So he is also asking when is your vaccine coming for clinical trials?
I said I cannot comment on that because I suppose not involved in the clinical studies and all those things because I'm a PhD. So the group is working, company is working, it will come. So it will come.
Okay. Thank you, sir. So the next question is from Soumya Ranjan. She has asked how the COVID patients are getting cured, by taking which drugs or therapies their body can generate antibodies to kill the virus.
Okay, keep in no therapeutic molecule does not generate antibody in your body. Keep in mind. So the drug you are taking is intervention. It's not like immunity. You are not involved in the immunity. Immunity is you have to generate immunity by throwing the antigens, right? Small molecules temporarily are blocking that interaction, so that does not going to generate immunity in your body, right? So each country, they're following the different rules. For example, some countries are using hydroxychloroquine for another drug. India also, like I heard, they're using, but I cannot comment on that because it's a physician level of discussion, so I should not talk. But each country they have, but there's no so far approved drug or anything for that COVID. But the anti-IL-6 inflammatory that also been saying that because sorry, the virus is not only infected the secondary infection, which means systemic cytokine storm, which is one of the key factor for a lot of death, right? So that's why they're using anti-IL-6 as a therapy. But again, it's a independent an individual country that make a decision, not there's no global recommendation from WHO or anything else. So I cannot comment on specifically.
Okay, thank you, sir. The next one is from Dr. Sharan Gowda. He has asked if vitamins and minerals are very significant in controlling COVID-19, why we need vaccine and can we go only with immunotherapy? What's your opinion?
Again, this is a physician question. So people, natural medicine, everybody's talking their own choice or preference, right? So as a scientific, I cannot comment on that, vitamin. If your data is there supporting that, yeah, of course you can do that. That's your choice. But anything you need to have a scientific data to support or prove your believeness. So I let it for the physician choice, not my choice.
Okay, thank you, sir. The next one is from Kasi Vishwanath. He has asked, according to you, what percentage the plasma therapy will work?
Yeah, that's a good. Plasma therapy will, because there's a lot of previous studies from not only COVID, other viruses or cancers and other things. Plasma therapy is one of the best way to stop. Not for 100% you can believe that it will work. So it's one of the good platform. But getting a right plasma purification, that costs a lot of money. How to deliver the amount, those kind of things take time. But plasma therapy is the best way to cure the infection.
Okay. Yeah. Thank you, sir. The next one is from Sashwat Kumar Mohanty. He has asked, are these vaccines developing B cells? As none of these vaccines showed the development of B cells, if not, then are these vaccines will boost the immune system for a long time? Will it be memorizing by our immune system?
I think she has to read So you have a naturally immune B cells and T cells, but they are not trained. That's what I said, put a slide, you need to educate them, right? Educate means, so our immune system is one of the world, like, I mean, the best thing, right? So, but they're called naive B cells, naive T cells, always there, right? When a foreign protein comes into your body, then your immune system trigger means activate. Then your B cells get activated. So there's not going to be it's necessary they will produce more B cells, but the B cell get knowledge. So sometimes the B cell doesn't get the knowledge, they will be silent. That's considered as a naive cells. But B cell, how they get, like, either they're getting one signal or two signal, multiple signal, depends upon they will develop. They will grow. Grow means mature. That's called mature. Once you get the mature B cells, that you'll lifelong until you die, that B cell will recall. Okay, that's why, like, best example I would say that, like, for example, chickenpox, smallpox vaccine, what we had, we never going to get back again the chickenpox infection or smallpox, right? So that's why that the B cells are trained. So B cells, you need to train them, or not only B cells, T cells also, both you need to train by vaccination. That's what the vaccine does that. But B cells always be there in your body, but they are naive. Naive means they don't know anything about it. Once they get the antigens or foreign protein, then they respond well.
Okay, thank you, sir. He also has another question. What is the possible chance of drug development for COVID?
Remember I said, I put a slide, like, there's a lot of drug is going on, small molecule, already used drug, like HIV drugs, like, because it's RNA virus, right? So they're trying to use that, that drug can be used for here. Studies going on right now. Already there's a lot of drug has been approved drug against RNA virus. They actually doing that studies going on right now, but no approved yet, but they are working on that.
Okay.
That may be a new drug also they're designing. I'm not expert on the drug development. So there is a lot of studies going
on. Okay. Yeah, thank you, sir. The next one is from Sudanan. He asked, why don't we
one more question. So can you ask one question from one volunteer, because I have to go another meeting. Like, is that right? We need to cover everyone, right? So one question from one.
Yeah. Yes. Yeah. So so can I ask, like, in total 5 questions or something like that?
No, you can ask. What I'm saying is, instead of asking a same person, right? So better to ask a single question from single. Right? One question from one person. Yes, please.
Yeah, sure. So now the question is, why don't we use hybridoma technology to produce large-scale antibodies by sequencing plasma cells that can produce effective antibodies against SARS-CoV-2 and the specific VDJ recombination sequence in them that can be genetically engineered to the hybridoma cells to create effective antibodies?
Yeah, all this antibody, like what we are, the one I presented, like, through hybridoma technology only, right? But the problem is, in order to generate hybridoma, it costs a lot of money because of purification process. It costs, like, for example, like anti-TNF alpha, put it that way. It costs like more than $5,000, like, for the single injection. Do you think in India, country like India or Africa can be able to afford, like poor people can afford that much money? That's what the synthetic DNA platform, what we are actually, after this hybridoma, we sequence the heavy chain, light chain. Like, we know this sequence, like, it works well. So we clone into our expression vector. Keep in mind, whoever the person, you can read my paper. I published like 3 papers on that platform. You can understand very clearly. So that DNA encoded antibody, when you inject into muscle cells, the muscle cells immediately produce protein, nothing but antibody. Keep in mind, because we are coating the heavy chain and light chain. That means immediately you will have an antibody systematically in your body within 12 hours. That's what the idea. So like a cheaper and easy way to generate a therapeutic interventions against the virus. So you can go on Google my name, like, you will see 2 papers on chikungunya and Zika we published recently. You can read that paper, you will get more information how this synthetic platform is, how great it is.
Okay.
But everything we are cloning from the hybridoma. Keep in mind, hybridoma. Without hybridoma.
Okay. Thank you, sir. The next one is from Dr. Sangeeta. She has asked, what is your opinion about Covaxin, a vaccine developed by Bharat Biotech in India?
Okay, it's good, but I need to wait and see the data. Like, without data, I cannot comment on anything, right? So I need to see the data. But the platform is really good. And also what I think I wasn't there yesterday in TV news also. Like, it's a good platform. Yeah, known platform. Inactivated vaccine is known because the rabies vaccine was developed from that same approach. So it's highly immunogenic. Like, highly definitely you will have a good response. But I don't have any data to comment on scientifically, so I cannot comment on that. So.
Okay, thank you, sir. The next one is from Barsha. SARS-CoV-19 is not the only coronavirus. There are other strains of coronavirus like OC43, 229E, NL63, HKU1, which most likely cause upper or lower respiratory infections. Can we use these strains for SARS-CoV vaccine production? Like, can we use them as live attenuated vaccine or chimera vaccine production?
It's a good question, actually. Thanks for that asking that question. Yes, coronavirus is like almost like 7 different viruses always be circulating in our body, like in our area environment, right? Yes, there's a studies going on. A lot of people are doing that, but the COVID, like a little bit changed from those virus. When you look at the genome of overall genome, the spike proteins somewhat different than that. That's why this is considered as a new virus, like COVID. Yes, a chimeric live attenuated may be a good platform. That studies also a lot of studies going on right now. Yes, it's a good question. Yeah.
Okay, thank you, sir. So the next one is related to plasma therapy from Paramita Sahu. In due course of time, the COVID-19 virus is developing different strains like L and S strain, which has been found in different areas. Then in this case, the use of plasma therapy can be effective, or in spite of that, any strain, plasma therapy is the best. You already answered that, but
yeah, plasma therapy is plasma therapy is like nothing but a plasma means it could be antibody therapy, it could be a purified from the patient, like a lot of plasma therapy is a big word, right? It's basically a monoclonal antibody, like antibody generated against the virus. So when you do that, like a mutation, that means like your immune system. So keep in mind, like when you are getting a mutation virus, forget about the mutation. If you are taking a plasma from a patient who recover from that infection, means they means they overcome that mutation, right? That means they also developed antibody against that mutation. Also keep in mind, right? So a lot of mutations, like right now in the last 2 weeks back in IIC Bangalore, like they said like more than like a 17 or 20 mutation in India. The circulation is going on. Yes, some of them is really bad. Some of them is like a normal mutation, so we don't need to worry about too much right now. But the vaccine is like again, it's a synthetic platform. You always change, you can modify that. But the concept is like the immunogen, like immunological response. Yes, vaccine is mutation is see, same thing. A flu always make a mutation. That's why every year like we are getting a new strain, right? Because RNA virus known to mutate. That's their job. Like how we are sneezing, same thing they are doing like a mutation. That's we cannot stop them. So yeah, plasma therapies. Keep in mind where you getting from this, what kind of patient. That means you have to go and sequence those patient, all the details, where are the mutation. Then you can apply. That's too much work. Like yeah.
Okay, thank you, sir. The next one is from Praveen Kumar. How cost effective is using monoclonal antibodies for therapy? Yeah, sure. Sorry. Take your time, sir. Take your time.
Yeah, so you see, monoclonal antibody therapy again is a problem. Not a problem, is cost-wise very expensive, like to make a recombinant antibody. But recombinant antibody is one of the successful therapeutic intervention for not only infectious disease, cancer, like autoimmune disease, like several cases, like a monoclonal antibody therapy is highly play a big role. Even autoimmune disease like Crohn's disease, rheumatoid arthritis, like now the only option is the antibody therapy. Antibody therapy is the best, but the problem is to synthesize or purification, a lot of involved, right? Overall, yes, in the future we may have a technology to speed up the process, reduce the cost. That's what I am saying, that synthetic DNA platform, which I mentioned, that we are cloning that particular good antibody clone, heavy chain, light chain, put them into expression vector. So when you inject it to your body itself make an antibody. So that cost only $10, for example. I am just giving a number. Then you inject your body, the body within a 12 hours to 24 hours, your body will produce that protein. That protein is nothing but antibody. So that's another approach is going on. A lot of studies going on. There is a phase 1 study started in US, for Zika studies. So Future, like there's a way you can generate an antibody. It's one of the best way for the therapeutic intervention. I would recommend strongly.
Okay. So there's another question from Pooja Ghosh. Why the symptoms varies among different person who gets infected?
Yeah, that's a common. It's a respiratory disease. Keep in mind, like best example, when you're staying in a home, okay, one person within your mom, sister, brother, uncle, whoever, one person get infection, rest of them recover, some of them get infected, right? It's a individual immune system how they're responding that viral disease. You may have an effect on the 100% response. Some people 50%. That individual response. So there's no scale for that. Like no, all people get disease. Like so, the natural like how immunity. Some people exposed to previously they may respond well, right? So that's a lot of factors. So there's no any common factor like why some people get not because of their immune system. How good, bad.
I think Dr. Subbalakshmi, you just limit the questions now and check with him how much time he is left with.
Yeah. Okay, sir.
I can talk another 10 more minutes. 10 more minutes. I have to leave by 11.
Okay, that is fine. Yeah. Otherwise, you can direct the questions to him later on in case they still are there. Okay.
Okay, sir. We will.
No, I can go another 5-7 minutes.
You can take other questions 5-10 minutes, no problem. But limit the questions because they cannot go endlessly.
Yeah.
So, don't ask the same question if there is like something interesting. Yeah.
So okay, I found another interesting question. So this is from Sri Vidya. Antibody titer decreases after peak of infection. Therefore, what is your opinion on whether antibody titer measurement is a reliable indicator to determine how long the immune response and protection against the virus lasts? Or does the long-term immunity to the virus mainly depend on long-lasting memory B cells? Why should we rely on more to predict immunity against viral infections?
I think question is like a complication of you're asking.
Yes.
Simple. The way actually I will rephrase it. Okay, antibody means you are talking about when you're in a chronic infection, the IgM, that's also antibody, right? IgM is a kind of pentamer, like keep in mind pentamer. IgM comes first, then remember one of the chart I showed, like then IgM goes down, then your IgG will come up, right? So IgG will depends upon how strong exposing. Keep on, for example, you are keep on exposing means that IgG will stay in your body. That's why the B cell get like now. I mean, like kind of like a punching, like no. So you're how many punching, like how can you withstand, right? The same thing, like now. So virus hitting you over and over, the B cell like resistant, then they become a good B cells, right? Because they are stronger, like they can be able to stop that. That's why the like constant exposure, because ongoing infection can leads to the B cell to mature more and more. So then they produce more antibody, they stay there. That again, individual to individual, it differ. Not we cannot, but that IgG once developed systematically, like then you will have a memory, like they will have the memory B cells. As long as you have a memory B cells, anytime when you come back that virus, same virus, they will immediately respond. So the measurement is like can differ from person to person, but that 15 days, like first 3 days like IgM, then they convert them to, that's called class switching, IgM to IgG. Something like in the mucosal area, like now it's become IgA, like the respiratory IgA, all the antibodies there. So antibodies is a big word, but there's like 5 different type of antibodies there.
Okay. So maybe we can wind up with this, or there's one more question if you would like to answer. Yeah. So can we implement direct cytotoxic T cell immunogenic therapy like we use in various cancer treatment in the COVID treatment in future? So can we use cytotoxic T cell immunogenic therapy for COVID? That's the question.
There's no way you can make a cytotoxic T cells in outside the body, right? You have to, right? You're talking about a CAR cell therapy. I think that maybe if I'm reading, it's a CAR cell therapy. That's a engineering T cells. So you have to take your T cells out, right, and engineering the T cells and put it back. That costs a lot of money, billions of dollars, I mean thousands of dollars. It's not possible, right? But there's engineering, like they're doing that, like in a different viral way can be able to generate. So that study is going on, but it takes some time. right now, like, there's a pandemic situation we are. Exploration is one way, like scientifically exploration one way. The same way, like you have to think about like economically and simplified, quickly how can deliver that vaccine. That's the very important key factor at the current situation, right? So we don't want to see. I have a lot of idea, but I cannot implement it. First of all, financial support, like because nobody really care about your exploration. Right now, we need a requirement. We need this done, right? Yes, there is a possibility you can engineering the T cells, which you could put it back to your immune system. But again, it needs more data to support that. Right? You for me to a student. I am a biochemistry student from Madurai. Yeah.
Okay. Okay, sir. Thank you very much for the excellent answers. Now I hand over the session to Dr. Rukmani. Thank you.
Yeah. Thank you very much, sir, for the mind-boggling question and answer session. We are extremely grateful to you for being with us, especially in the early hours of your day. Thank you so much, sir.
Thank you. Thanks for all. Thank you. Sorry, I had to go because Friday, like I have 2 more meetings. I need to go. Thanks.
Now I request Dr. Sudharani Madam to propose vote of thanks. Yeah, with that.
To Dr. Rukmani, on behalf of the organizing committee of the week-long international webinar, Covinar, I profusely thank our speaker of today's session, Professor Kar Muthumani from Western Institute, for his highly informative lecture. Thank you, sir, for explaining in detail on the coronavirus infections and about different types of vaccines developed for coronavirus 2, and also throwing light on their immunological reactions during preclinical and clinical trials. And you also spoke on the possible therapeutic interventions and their uses. Thank you very much, sir, also for clearing the doubts regarding the vaccines and the potential use of therapeutic interventions. And I am very sure that this talk has immensely helped all the participants. Thank you very much, sir.
Thank you for all. Stay safe. Hopefully, we will get something soon. We will all become a normal, like you, right? See you guys.
Thank you.
Thank you, sir.
Thank you.
Juncture, I also thank, I take this opportunity to thank our Vice Chancellor, Professor Gurmeet Singh, for his unstinted support and encouragement. I must also thank Professor Balakrishnan, our Director, SAIRR, for his support. And my sincere thanks to Professor P.P. Mathur, our HOD and Dean School of Life Sciences, who is the motivating force in organizing this Covinar, the week-long webinar Covinar. Thank you very much, sir. And
thank you.
Definitely thank all the speakers of this webinar who accepted in very short notice. The speaker for the first day was Dr. Pavitra Venkatagopalan from Care Health Diagnostics, Chennai, who spoke on molecular biology and life cycle of coronaviruses, which was very interesting and welcomed by all the participants. The second day's speech was given by Professor V. Gopal from Mother Teresa Institute of Health Sciences, Pondicherry. He spoke on traditional medicines for COVID-19 infections. And on the third day, we had the speech from Professor Sishubala from Davangere University, and he spoke on role of immunity in COVID-19, which was an interesting talk. And on the fourth day, Dr. Vijay P. Singh from Mayo Clinic, Arizona, USA, he spoke on lipotoxic exacerbations of COVID-19 and simple interventions to improve the outcomes of COVID-19 infections, which was also very interesting. So I also thank all the participants for their active participation and cooperation during this week-long webinar for this whole week. We had more than 1,100 registrations, and we had participants from all over the country and also from USA, Africa, Middle East, and Norwegian countries. And all the participants interacted well with the speakers and showed much interest in the whole session. Thank you, all the participants. I thank the Computer Center for their support. And I must thank all the PhD students and MSc students of our department who gave their complete support in all the academic activities of the department, and also in this webinar, organizing this webinar also. My special thanks to Akshaya and Abhishek, our MSc students, who coined the term Covinar for this webinar. Thank you, students. And last but not the least, I must thank Dr. Balaji Boragwan and Mr. Abhishek for their technical support, without whose support this webinar would not have become possible. Thank you all. Thank you very much.
Thank you very much, madam. I thank all the participants for your enthusiasm, continuous support, and cooperation throughout the webinar. The link for feedback form and the other supporting materials are sent to your registered mail ID, and it is also given in the chat box. You can directly download your certificate after filling the feedback form. The feedback form will be open only now. Please don't worry if it was not working before. And if you have any problem in downloading the certificate, please contact us or try after 24 hours. Thank you very much for your support once again, and all the very best. Together, let's all strive for the COVID-free world. Thank you very much, all of you.
Thank you.
Okay. Thank you, Doctor.
Thank you. Thank you very much all. Bye-bye.